Thank you, everyone. So my name is Davide Danovi. It's a pleasure to be here. For those of you who are coming straight from the stem cells at lunch, it will be like a marathon. I will try to give you a sense of what our interests are in terms of using high content to characterize stem cells. So in the first part, I will overview some of the theory of it, and I will present some data from HIPSI that's already published. In the second part, I will focus on a recent project, and in the third part, I will give you a sense of our facility, the Stem Cell Hotel, that's being relaunching very soon. So while we're all busy with our favorite activities, the world is changing. There is a clear sense, for example, that from images, we can extract a lot of information and a lot of data, and also visualization of data can turn into image at the same time. If you look at a map that represents the flights in the beginning of the year, you will think that in the southeast of this island, there is a big city. Probably if you were to look at this same map in more recent times, you would see that something is missing. What do we miss the most in terms of the activities we have in the lab? I think the scientific community is largely swaying at this point in time between people who are focused on an old model, if you wish, of generating an hypothesis, testing an hypothesis, versus a new trend that is emerging where hypotheses are directly linked to the data. And so there was an interesting commentary in Nature a few years ago about people who were pushing for or against these points. And I think there is a constant tension between these two type of work, but actually there are ways to reconcile them. The type of work has profound implications in the type of data you're collecting. So I think it's really emerging as a great theme that one needs to know exactly what type of information we're gathering from what type of experiments. And this is an example taken from the Human Cell Atlas. But it also has profound implication of how we fund the production of science and how we communicate the results. And so for those interested in these topics, I've written a small book chapter with a colleague of mine, and I'd be very happy to talk about this. But in terms of what we do in the lab, we use cell imaging to benchmark human cells. And so we're very interested in using stem cells, in particular induced pluripotent stem cells. The promise of induced pluripotent stem cells, like most people here would know, is to be able to have an embryonic-like cells from a differentiated cell in the human body, and to use this for disease modeling, for drug discovery, and for cell therapy. What we're very, very interested in is to be able to define and quantify the variance in the cells that we extract from the images. And in particular, we've been really committed in identifying and quantifying the variance that comes from different donors versus the variance that comes from the different environment the cells are in, the conditions the cells are cultured in. So we do this by using high content analysis. This is an example of an image taken from our Operetta device. We can segment the cells, we can identify the nuclei, identify markers like EDU for proliferation, for example. And then we can use cell mask to understand the boundaries of a cell. We find the cells. We can use context features such as how many cells are present in a colony that give us an example of an idea of what cell adhesion property each cell has. And this was really set up in the beginning as a way to collate the information from the cell images with data that comes from other biological data sets. So within the framework of the Human-Induced Pluripotent Stem Cell Initiative, we have been characterizing the cell images and collating the data with the help of EBI to the genomics data from the Sanger and the proteomics data from Dundee. And so what I really loved about this project, and it was a great privilege to be able to take this project to completion with the rest of the team, is that it really sits in between the possibility of having reductionist type data sets that are accessible to the scientific community and bridging these data sets with the biobanking. So the scientists can access data regarding each cell lines, but they also can order the cell lines from the repository. And also our images are now available in the image data resource by Jason Swedler in Dundee, run by Jason Swedler in Dundee. And this is really possibility to engage other people that are interested in the analysis in the effort to collate the data with the genomics and to understand the particular relationship between this. So I won't spend much about this work. I will just refer to the publication. The publication in Nature in 2017 gave a sort of overview of the project, and we included data from a few donors for which we had clones for 2 lines. We went much deeper into this analysis in recent times with the help of Alessandra Vigilante, who has been driving this effort. And we have analyzed, in this case, 110 induced pluripotent stem cell lines derived from 75 healthy individuals. And in terms of deliverable, this study gave a sense of a method to analyze these images, the possibility of using multidimensional reduction to capture parameters that were related to the donors versus parameters that were related to the extrinsic environment, and then naive forays into the association between the genomics and the phenotyping by way of identifying phenotypic outlier and matching these to single nucleotide variants. So in essence, we have developed a set of tools that allow us to evaluate this. We've also worked on the dynamics, and I'm not mentioning this here. We have managed to demonstrate that genetics is responsible for a portion of this variance, and we have started to define associations between particular genes and particular phenotype. So in the second part of the talk, in the core of my little chat today, I would focus on a project that has been published very recently, and it's mainly been driven by Hanin Al-Sali in the lab. So we've decided to develop the type of methods array and to extend the readout from single 2D endpoint, single cell 2D endpoint images into the possibility of tapping into more complex environment. So there's work going on the so-called 2.5D type of environments. So for example, in this case, you can see work mainly driven by Alice Vickers and Mukul Tewari, where there was a possibility to confine in a 1 millimeter pattern iPS cells. And what I will focus in the next few slides is our work aimed at defining 3D images from iPS cells. So Hanin has basically taken iPS cells and cultured them in E8 medium in self-renewing condition, but in suspension. So the cells are forming spheroids. And at day 2, these spheroids are transferred in 2 different type of conditions. They are either kept in E8 for further expansion, or they are moved still in suspension to a condition that pushes differentiation, so KSR and BMP4. And throughout this process, we have taken time frame images, and at the end, we observe that the shape of the spheroid is very different in this condition, and the differentiation also happens. So if you look at the images throughout time, you can see, and also the consistency of this, you can see that at the end of this process, you have consistently very round spheroids in the E8 medium and differentiated cells in the KSR plus BMP4. So here you can see this in live images. You have a clear round spheroid emerging from the self-renewing condition, and you have a more differentiating condition that triggers a shape that is less symmetrical. It's very asymmetric, and you have budding and different formation. So it's quite challenging to quantify this and to segment these images. So with the help of Fuad Moses in the lab, we managed to use cell profiler pipelines. And the trick here was to subtract an image from the following hour in terms of pixel intensities, so that we were left simply with the changes in between these images, so the delta between these images. And when you segment this as an object in its own right, you can capture much better the type of spheroids. So we've analyzed this, and we've also run this with the help of micrographia using convolutional neural network, with the idea of predicting the conditions that would give rise to a budded spheroid or a round spheroid. And you can see on the right side of the slide that the accuracy of prediction of this method gets better and better over time. It's almost random in the initial frames, but it becomes much more efficient, much more accurate towards the end of the process. So this leaves us now with the possibility of matching this live imaging in blurry images, so to speak, but dynamic, with a much more detailed type of imaging that you can obtain from the endpoint. And this has always been, also in my previous work, something very close to my heart, to sort of try to match these two things. And in this particular spheroids, we are observing emergence of differentiated lineages and reduction of the pluripotent stem cell markers. So the question that Hanini is now trying to tackle is how this live image relates with the endpoint. And also in collaboration with Aline Gentleman, we are trying to culture these cells not in suspension, but in defined gels with defined properties, so that we could counteract this morphogenesis events and ask the question of whether we would still observe the differentiated cells from this. So I would recommend anybody who would like to who has kind of a background in this respect, to get in touch with us and to speak with Aline. And we are now driving this method into the biology, understanding differentiation and morphogenesis as separate event. In the final part of my talk, I would give you a sense of how we really feel this method can be used in different directions towards translational application. And so in terms of the boundaries of the cells, we have worked with the Lorenzo Veschini's group, and we published last year a paper where we've been observing, in particular, the junctions of endothelial cells. And the question here is to define how different endothelial cells that are derived from IPS are from primary human endothelial cells. And we were able to use these methods for cell imaging, but also methods that now tackle the actual boundaries of a cell in order to address this and quantify these differences. And also we are working very actively in a different cell type using organoid from IPS derived neural cells, and in co-culture with patient-derived brain tumor cells, we are now imaging how the migration of brain tumor cells happens on the axon. And we are using this in particular for the project of Victoria Tsang in the lab, who's a Wellcome Trust PhD student, with the aim of defining complex environment that could be used to test the migration of the cells. She has been using the quantitative phase imaging devices, you can see on the top left, to identify the objects and evaluate the migration in different types of context in a precise way without any fluorescent markers. So this leads me to the end of my talk, and really the sense is that this capability that was born within the framework of HIPSI and is now being refined into addressing different types of biological questions is also provided as a service for fruition to users. So we have 5 devices for high continent imaging, and we are able to assist the users and to provide expertise. And I'm a strong believer in this model. It optimizes the possibility of using the devices. It provides opportunity to link with academia and biotech, and it really pushes the development of new methods forward. So we had recently a grant from MedCity, for example, with a company called Lift that is coming to the Stem Cell Hotel in order to refine some of their cell therapy product. And we have a case studentship with Orchard and an MRC program grant that is aiming at defining the susceptibility to viral infection, HIV-1 and HIV-2, across the population of HIPSI cell lines. And also we have a collaboration that is coming soon where a Danish company, Celari, is hosting our server, and we allow a citizen science project aiming at segmenting cells in order to establish a ground truth to develop AI algorithms that are more precise for cell segmentation. And just to give you a sense of how broad these collaborations are, I was recently privileged to be joining a consortium that is led by OpenCell that is developing analytics for a shipping container-based modular lab. And this would be potentially a solution that can reach areas that are otherwise hours away from a testing center without any need to ship any material. And with the OpenTrons robot inside, it can give up to 2,000 tests a day. So the innovation, the service, and the research aspect, I think culturally are in silos, and this is to some extent an inefficiency of the system. I think the data-driven approaches can synergize with the hypothesis-driven research, and also the different type of communities can really merge effectively. So we are interested in developing the methods, and we are working actively with the technology providers, such as the projects I mentioned in collaboration with PerkinElmer, Dynamics, Solari, and Micrographia. And we're also now more and more getting companies to engage with us from the point of view of actual deliverable that are required for their progress. So the possibility of the London Stem Cell Network, and I'm very grateful for being hosted here today, and it's very close to my heart because we've been working in really establishing a community of stem cell scientists in London that can share expertise. There are other communities that I really would invite the audience to consider. One is the computer-aided biology community that has at its core this idea of merging the hypothesis-driven and the data-driven research. And I was very recently happy to join this incredible project from the Academy of Medical Science, which is called Flyers, a Future Leader, Innovation, Enterprise, and Research, which is really aiming to sort of foster a culture of cross-sector development. And I think one shouldn't be doing everything, but it's really good to be able to bridge the different sectors in a way that allows research to be finding a translational application. We hope we could give you a sense of our activities, and really the aim for the Stem Cell Hotel is to become a nucleus for this collaborative phenotyping and reaching out at these different type of communities. Thank you very much.